Okay, so now we're going to talk about temperature limits in aquatic life. And we're going to first talk about heat. What is heat? Like, if you're thinking in the sense of physics, what is heat? Heat is energy. It is kinetic energy. It is energy that makes stuff bounce and go crazy, right? And so as you increase the heat in an area, you're going to get more movement in those molecules. And so you're going to see temperature influencing a lot of physiological processes. Typically, as temperature goes up, all your metabolic processes go up. So you have faster respiration, you'll use more oxygen, you'll make stuff quicker, faster, bigger, all with an increase of heat to a point. Once you get around 50 degrees Celsius, in biological systems, stuff starts breaking down. That's because you have these parts, and now they're bouncing all over the place, and they'll start to break. Proteins will lose their shape, something called denaturing. But in normal circumstances, it typically doesn't get up to 50 degrees Celsius. Occasionally, like desert sands or like a place like a geyser or those underwater, those deep sea vents, like they'll get it pretty hot there. Typically it doesn't get that hot. And we can measure, or we have a measure, of how much 10 degrees change will influence an organism's function. And so you'll pick some certain physiological function, whatever it is, and you'll measure it at, in this case, 25, and then you measure it again at 15, and you get up this number called the Q10 ratio. So your Q10 is going to be whatever that physiological process is, the ratio at the two different degrees with the hotter degree on top. So in this example, we're using oxygen use, and so how much O2 do they consume? As it goes up, you see it increases for this set value. So the Q10 for the oxygen use in this organism would be, what is this, like around 600 divided by 350, give or take. So that gives us your Q10 value. Some animals like it really hot. These are called thermophilic organisms. I say animals. Organisms. Or heat-loving. Philos means to love. Thermos, talking about the heat. Typically, you don't get organisms in temperatures that reach up to like 50 degrees Celsius. But occasionally, you do. So at these hot springs, you can see there's all these really cool colors going on here. These cool colors are due to bacteria. These bacteria can live in the hot springs. They're thermophilic. They really, really like it. And they actually, studying these bacteria helped genetics a ton. So when scientists were figuring out how do these bacteria live in this really hot environment without just denaturing everything, well, they took them and they found that their DNA polymerase works under high temperatures. Typically under high temperatures, DNA splits apart. And so we were able to develop this technique called PCR, polymerase chain reaction. Maybe you've heard of it. It's a really big deal, a great way to amplify lots of DNA really quickly. We're able to use their DNA polymerase because it doesn't denature at high temperatures. And so as you heat it up, the DNA falls apart, the polymerase hooks on with a primer, copies the DNA, you make a copy, put it back together, pull it back apart, make another copy, and you just do that over and over. And you're able to get this logarithmic growth from or of DNA. It's really cool, really useful. And something that we learned from thermophilic organisms. Very cool. So temperature doesn't normally get above boiling. It doesn't normally, or even half boiling. It doesn't typically get above 50 degrees Celsius. But it very often gets below freezing. What happens when stuff gets below freezing? Well, water freezes, right? How much water is in you? Depending on what you hear, 90-something% water, right? There's a lot of water in your cells, outside of your cells, everywhere in your body. So you can walk outside when it's freezing and you don't freeze, right? Because we have mechanisms that protect us from that. We are homeotherms, so we produce our own heat. We have behaviors like wrapping up in clothes that help stop you from freezing. But what happens if the water in your body does start to freeze? Well, it's going to start freezing around this thing called a seed. Typically, it's like an ice crystal that's already formed on something else, and then it just expands out from there. And when it does that, it's going to damage the cells, it's going to destroy organelles, it's going to just make a mess of everything. If you've ever seen frostbite, that's what has happened. A lot of our aquatic animals live in stuff that is below 0 degrees. So seawater, it freezes at negative 1.9 degrees. So it actually freezes below 0. The water inside the animals will freeze close to 0 if they didn't have ways to stop that. So if you just take an animal that's not adapted for the cold, put them in the cold, they will freeze solid. So what are some of the ways that they protect against freezing? So one way they can do it is by putting glycerol in their blood. So glycerol is a sugar, and it prevents these hydrogen bonds from forming and sticking and freezing together. So it decreases the freezing temperature. Also, glycoproteins do pretty much the same job. They lower these temperatures. So Antarctic fish, like these cod, can handle very cold, that's ice right there, can handle very cold temperatures because they have glycerol in their body. Glycerol, just a 10% increase of glycerol, or 10% solution of glycerol in the body, can decrease the freezing temperature to negative 2.3 degrees. And if you remember, it freezes at negative 1.9. So if it makes it down to negative 2.3, you're safe. You're not going to freeze before the water freezes. Now what happens if the water freezes? Well, now we can have some issues, right? But this glycerol and these glycoproteins act like an antifreeze, and in fact, they're pretty much the same stuff that you do find in antifreeze. Another way organisms can survive the cold is by supercooling. What happens is these glycoproteins stop ice formation by coating any ice crystals they find. So if they find an ice crystal, they coat it, and what causes ice to grow are those seeds, are these little ice crystals, and then the ice grows out from that. And so that can lower the freezing temperature. It's been seen in reptiles, I think, down to like negative 8 degrees Celsius, stopping the ice formation, and around negative 16 in amphibians. And so the supercooling can work really well. What happens if it's not super cold or super hot? Well, even if it is super cold, as long as that animal is adapted to it, it'll be alright. Or if it's super hot, it'll be alright. Because life has a thermal optima. It has a range in which it works best. And in a lot of places, especially temperate climates, you're going to see fluctuations, right? In winter, it's going to be cold. In summer, it's going to be hot. how can an animal be optimal in both cold and hot temperatures? Well, they have isozymes. An isozyme is an enzyme that has two different forms. And so you can see here that in this fish, you're going to have an isozyme that is active during the winter, and its activity works really good in low temperatures. As it gets warmer, its activity drops off. But it has the same enzyme in a different shape that works really good at warm temperatures. And so with these various enzymes, with these isozymes, basically doing the same job, just a different structure, because it performs good at one temperature and not the other. An animal is able to survive and thrive in large, different temperature environments. If it's not a very different temperature environment, then they'll probably just have one enzyme that does that job. But if it is really varied, then they can have two. So, what is an adaptation that helps thermophiles in high temperatures? You can think of that DNA polymerase that you find in bacteria, the tadpole. Describe some adaptations that enable fish to survive in very cold water. Talking about antifreeze. And then how do isozymes help organism functions in a large range of temperatures? I don't know what, in a side range of temperatures. In a large range of temperatures. And so they're able to share that, right? So in cold you'll have one, and in warm you'll have another enzyme. And with that, we are done talking about water.